We will now travel from Italy eastward to the fine city of Brno in Czechia. And we will have another node and user presentation. Martin Gajdoš from CEITEC Masaryk University and Michal Mikl at the ALM and the medical imaging node in Brno will present how to identify epileptogenic zone in MRI negative epilepsy with arterial spin labeling data.
So, and in the next few minutes, I will talk about epilepsy, about non-lesional epilepsy and arterial spin labeling data and what we measured here in Brno. To introduce myself, I'm a junior researcher in multimodal neuroimaging research group at CEITEC in Brno in Czech Republic. So my talk will be about epilepsy and arterial spin labeling and about a developed method for use of arterial spin labeling data for identification of epileptogenic zone. So probably all of you are aware of epilepsy, which is a neurological disease with a prevalence in population about 1%. The usual diagnostics for epilepsy is ictal and interictal EEG and video EEG. Usually also anatomical MRI, PET and SPECT are also used. And experimentally, you can use, for example, arterial spin labeling data where you are interested in interictal hypoperfusion. Here on the figure, you can see EEG recording from high-density EEG of an epileptogenic spike. And here is a reconstruction of such spike in source. So this is a special technique called electrical source imaging. So about 30% of epilepsy patients do not respond to drugs, to anti-epileptic medication, and they are optimal candidates for surgery. And unfortunately, in about 20 to 40 epilepsy surgery candidates, there is no identifiable epileptogenic lesion which is detectable with standard diagnostic like anatomical MRI. And sometimes the diagnostic methods are in divergence. So in this case, the patient is called non-lesional, and it's more difficult to help him, for example, with epilepsy surgery. Arterial spin labeling is MR technique, which is perfect for measurement of perfusion. The pipeline consists of tagging the blood inflowing into head. So there is one image where we use this radio frequency tagging. We actually use the water in blood as natural tracer. And then we record the data. In next step, we repeat the experiment without this tagging, and we acquire the control image. And then we have we perform subtraction of control and tag images. And this difference in magnetization between control and tag conditions is then proportional to regional cerebral blood flow. So now some terminology. With ASL, we are able to imagine the brain This part of head we use for labeling the blood. And in our case, we used pseudo-continuous ASL, which is abbreviated with PCASL. And one repetition time has several parts. It's labeling duration. This is the time where we label the blood. Then there is a post-label delay, the time when we wait until the labeled blood enters the brain, and then we acquire the image. There is one very nice review on the method. If somebody of you is interested, I depicted here. So, and what we measured in Brno and Mafil, we measured 29 patients with non-lesional epilepsy. Out of them, 7 were in a post-operative group. That means they were after successful surgery, and 22 out of them were in pre-surgery group, waiting for the surgery, but with a strong hypothesis about epileptogenic zone. Our aim was to use this ASL data to identify the epileptogenic zone. In post-operative group, we had resection mask for verification purposes. In preoperative group, we had mask of hypothesis, which was bigger, but also we could use it for verification. The ASL data consists of 41 pairs of control and labeled images, and we'll acquired also the N0 scan, which is something like a background noise, and the perfusion acquisition took about 6 minutes. Then we acquired T1 anatomical images to have good knowledge about anatomy of the patient's brain. Now, to have imagination how such imaging looks like. This is a T1 image where you can see relatively nice anatomy. And these are the ASL images. This is the ASL image labeled with labeled blood. This is without labeled blood, and this is the background. At the first look, you cannot see any differences with your own eyes. There we need math to see the differences. With our data, we used the tool Explore ASL pipeline, which was published, and our preprocessing consisted of a few steps. We corrected the scans for motion. We excluded scans with excessive movement. We quantified the cerebral blood flow based on this formula. Almost everything in the formula are constants beside of this SI control, which is for control image, SI label is for label images, and SIPD is for the background, for the ASL background image. Then we performed partial volume correction and computed asymmetry index. The asymmetry index is computed from the voxels of cerebral blood flow of one side of the hemisphere and subtracted with the voxel which is on the opposite part of the brain, on the opposite hemisphere. Next step was spatial smoothing with Gaussian kernel and masking with a priori mask of intracranial volume. So this was a lot of numbers and letters. So if we have look on the images, If we subtract the control and label image, we get perfusion weighted image, which looks like this. After applying the quantification formula, we get cerebral blood flow images, which look like this. And the advantage of this image is that it is in exact values. It's volume per amount of tissue in time. Out of these values, we can calculate the asymmetry index, which is depicted here. And we used only negative values because in perfusion, we are searching for interictal hypoperfusion, which means the place where which is probable epileptogenic zone is in the time when there is no ictus in hypoperfusion. So, and as a spoiler, there is anatomic image of the patient with a resection mask. So you can see there is some identity of this area and this area, and I will show it in next slides. There are also published methods for where the teams needed ASL data for detection of epileptogenic zone. We found two methods from team of Galazzo and from team of Koyan. The Galazzo team used a comparison of non-lesional epilepsy, a patient with non-lesional epilepsy with healthy control. And they used also asymmetric index data, and they used thresholding based on Z values greater than 1.64, which corresponds to t-test with p-value 0.05. The team of Koyan used thresholding with 95 percentile value of the patient's asymmetry index. Our aim was to use adaptive thresholding, and our method we developed was published last year in scientific reports. We used the histogram of asymmetry index values and normalized the values. In next step, we fitted a curve, exponential curve, and then we were interested in some interesting points of the curve. We identified the points of minimal distance to 0.00, which is here marked with a red line. We identified the elbow point where the exponential curve bends. And we identified the minimal product criterion as a minimal product of normalized counts and asymmetry indices, here marked with a black line. Based on these thresholds, we get clusters and evaluated true positive and false positive rates based on resection mask and based on hypothesis mask. We also evaluate positive predictive value. This was the most important metric for us. And here on the image, you can see slice view where in the rows is the resection mask, and other colors are clusters which arise after application of the minimum product criterion. So you can see there are some clusters which are not present in the resection mask, but importantly, there is a big cluster overlapping with the resection mask. Now with the different presentation, Here you see bigger image with a resection mask of one selected patient from our data set. And here you can see a thresholded asymmetry index map. And the important thing is a nice overlap of the resection mask with the thresholded asymmetry index mask. So as our result of our study was that the minimal product criterion outperformed the other two criterions we were testing in almost all of observed matrices. And the most important, it was the best criterion for the positive predictive value in both groups, in the preoperative group and also in the post-operative group. The minimal product criterion also outperformed the methods used with the team of Galazzo and Koyan, and most importantly in the positive predictive value. So here are the values or the results with usage of already published methods. Here is the developed method. So to sum up and to discuss, the calculation of the asymmetry index is not computationally demanding. So it could be implemented within a radiological viewer on MR scanners. The advantage of our method is that it's independent on a priori hypothesis about some parcellation schemes. It's independent on ROI-based analysis. And it doesn't require data from healthy controls as a calibration group, because this could be an obstacle if different groups and hospitals would like to use the same approach. They would need to acquire the data from healthy controls. Disadvantage of this approach is inability to detect symmetrical disruptions in perfusion. This is due to use of asymmetry index. Another important message is that arterial spin labeling is very helpful in combination with other imaging methods for epilepsy zone detection. And there is one nice paper from a colleague Masaryk, which was published last year. For those of you who are interested in references, here are they. And I would like to thank you very much for your attention. And I also want to many thanks to core facility MAFIL in Brno for the high quality measurement and for very useful assistance, which was crucial for this publication. So
many thanks, Martin. And
yeah.
Was it so that Michal, you wanted to say a few words, or?
Yes, good afternoon, everybody. I didn't prepare a specific promotion of the whole node or our lab, just a few comments to this project and maybe some general comments to similar type of services we can provide. So this was a nice example of a project, or maybe not just one project, but some combination of long-term activities in the study of epilepsy, which is coordinated by Professor Rector or by Professor Brazdil. It's an example of project which is done in collaboration between Masaryk University, the CEITEC, medical faculty, and university hospitals in Brno. And the measurements which Martin presented, or some of these measurements as output of his work, started in 2016, and to these days there was more than 700 hours of MRI measurements and 400 hours of EEG measurements, some simultaneous EEG fMRI or standalone EEG measured for these epileptic projects, and many patients and also healthy controls were measured, but because of necessity of some gold standard for evaluation of techniques method developed in these projects, only about 40 patients, which some surgery outcome was used in these studies, and maybe others will be used in other evaluations in subsequent projects. So here, Martin presented arterial spin labeling as a method of perfusion, but in this project, a wide range of MRI techniques was used, also some clinical part of protocol for epilepsy patients, then high resolution structure data for brain morphometry, both T1 and T2 weighted, diffusion weighted imaging, resting state fMRI, and other measurements. So it's also example of project where the total measurement time was about 2 hours. It was split into sessions. In one of these sessions, simultaneous EEG fMRI was measured. So it's example of project we can do here in Brno. Also similar projects, some long-term projects are done here with respect to neurodegenerative disease, Alzheimer's disease, Parkinson's disease, typically coordinated by Professor Rektorová, which are typically lasting for 3 or 4 years because it's necessary to find the patients and do all the examinations. So that's probably all from my side. Thank you.
Excellent and fantastic development again.